So we talked about in my last lecture on how structure is important to function, and that the membrane is going to be a really important example of that. So let me give you one piece of what I mean. And so remember these guys here, these guys represent your phospholipids. But at one point early in the semester, we actually drew a phospholipid in excruciating detail. And remember what we drew was we drew a 3-carbon chain that had an alcohol group, all right, on this end, and then hydrogen surrounding the rest of it. And we called that an alcohol because it had that OH group. And specifically, this was 1,2,3 propane triol. That was its name. The other name for it is glycerol, okay? Well, then we talked about how you could take a fatty acid chain that had a double bonded oxygen and an OH group, and then a long series of carbons, right, down here, and you could link this fatty acid chain to this glycerol by removing a water molecule, okay? And we called it a condensation reaction because the water would be a byproduct of the reaction. And so you separate, all right, you separate that OH group and that hydrogen there, you separate that water molecule, and what you get is you get this oxygen now bonded to that carbon, and you get the beginning of a phospholipid, okay? If we did that again over here off this guy, then we'd have another carbon, double bonded oxygen, and all these carbon chains, but this guy is going to have a double bond, which is going to cause this kind of kink in the tail here, okay? Okay, so now I've got 2 fatty acids, 2 phospholipids. This guy gets reverted, the oxygen goes up, and now it connects to a phosphate group along the top, okay? And so what we have is we have a phospholipid made up of 2 fatty acids and a phosphate group, okay? The head of this molecule up here, because of all of those oxygens, remember oxygen was highly electronegative. Oxygen is an electron hog. And so because the oxygen's sticking off the sides of this molecule, it would create some polarity. And so we say the head, all right, of this molecule is polar, all right? The head region is polar. Now, the tails of this molecule, these 2 long tails, remember they are surrounded by hydrogens. And hydrogens surrounding carbons tend to be nonpolar because each of those hydrogens is pulling electrons with the same strength, and so there's no negative or positive side, no polarity to the tails, all right? They are essentially nonpolar. And so that's kind of what we say. We say the tails of phospholipids are nonpolar, okay? And so we'll, draw that in as well. And so the tails are nonpolar. All right? Now, additionally, you have 2 tails. One is completely filled with hydrogen because there's no double bonds between the carbon. And if I'm completely filled with hydrogen, I am a saturated fatty acid, saturated with hydrogen. This one, on the other hand, is a double bond, so it is an unsaturated fatty acid because it has a double bond which removes hydrogens. It means there's less hydrogen than it could possibly hold, so it's unsaturated, okay? Now, if you remember, saturated fats tend to be solid at room temperature, but unsaturated fats tend to be liquid at room temperature. And so now we're going to talk about, now I just got done talking about the structure of a phospholipid, now let's talk about its function, okay? First, it's a barrier to polar ions, right, and other molecules. Well, how is it a barrier? Well, if we know that these tails are non-polar, okay, then we look in here and go, oh, well, this whole region right there, that tail region, is going to be non-polar. The other word for that is hydrophobic, okay? Doesn't like water. And anything like water, anything that's polar, would be repelled by that non-polar region. And so its structure is comprised of these long non-polar tails, which affect its function, which is to create a non-polar region and block polar molecules from being able to get in and out of the cell. Additionally, all these phospholipids have to be linked together to form a barrier or a membrane. Well, the head region, because it's polar, has negative side and positive side. So you get negative, positive, negative, positive, negative, positive, and the head region ends up being a magnet that allows all of these guys to line up and create that nice barrier. And so the head region being polar creates this magnetic barrier across the whole membrane. So again, structure to function. Lastly, this guy is meant to be somewhat solid. It's meant to be a membrane that is selectively permeable. So it blocks some things from going in and out, but it will allow other things to go in and out of the cell. So that would require it to be not too solid, but also not too liquid or watery, because if it was too liquidy, then it would dissolve and fall apart, it would break apart, and it wouldn't form a good barrier. So by having a saturated tail that's normally solid at room temperature, and an unsaturated tail that's normally liquid at room temperature, I'm not too solid, I'm not too liquid, but I'm this perfect balance that allows for the membrane to be selectively permeable, okay? So again, 3 examples there where we have structure determining function in excruciating detail with our help with understanding the structure of a phospholipid and how that would influence how the phospholipids function to help build that selectively permeable membrane, okay? Additional structure and function. The shape of this receptor is going to determine what it's able to grab a hold of. Think of that receptor as kind of like the active site of an enzyme. And so its shape is going to allow it to receive a molecule. If that shape changes, it's going to do a different job, okay? If it was a mutation, it might not do its job well, and then that would be a problem. If it was just an allele change, then it would just change what that receptor does to receive a different signal. Markers, their shape determines function as well, because if I change the shape of that marker, then I'm going to communicate differently to other cells. One example of a marker protein is the ABO blood group, right? A is a particular shaped marker on red blood cells. B is a different shaped marker. And so if I'm type A blood, but somebody gives me type B blood, because of the difference in shape of that marker, my body would attack it because my immune system doesn't recognize type B blood. And it would attack it, form a blood clot, and I could potentially die from that, okay? So, just wanted to go a little deeper into how the structure determines function.